Welcome to the Lab of Ornithology. My name is Lisa Kopp, and I am the Visitor Experience Manager here at the Lab of Ornithology. And I'm so happy to see you all here this evening, and I want to welcome the live stream audience that is tuning in from all over the world, potentially. So I want to get through a couple quick announcements before we get started with tonight's presentation. And the first is to talk about our upcoming Monday night seminar, which is going to be April 25th. Right here at the lab at 7:30 p.m. And that is going to be featuring author Daryl McGrath, who just published a book called Flight Paths, a field journey of hope, heartbreak, and miracles with New York's bird people. And she'll be speaking about the role that women biologists specifically played in saving the peregrine falcon and bald eagle. So that should be a very special presentation. Be sure to tune in for that. But tonight we have something special and unique. You'll be hearing from 3 ornithologists relatively new to the field. And some of you know that the lab has a rich history of working with students at all levels. And we've got a really special evening tonight because you're going to hear from someone at each of those 3 groups, an undergraduate, a graduate student, and a postdoc. So our first speaker tonight is Taylor Heaton Crisologo. Taylor is an undergraduate student studying ecology and evolutionary biology. She's spent 2 summers off the coast of Maine working on herring gull nest defense behavior, which you'll hear about today, and spent last summer in Australia working with superb lyrebirds in the Blue Mountains. After earning her undergraduate degree this spring, she plans to return to Australia to work as a field tech for a project on red-backed fairy wrens. And while she's there, she plans to visit and check out Australian graduate schools. So welcome to Taylor.
Thank you. I'd like to begin my presentation with a quote. To a hasty visitor, a herring gull colony is nothing more than a noisy place. All he remembers afterwards is a chaotic mass of screaming gulls, all flying excitedly round, and occasionally swooping down and delivering either a blow or a load of warm, smelly, and sticky substance. To the patient watcher, it is different. This is from the book Herring Gulls World, written by Nico Tinbergen, the father of modern ethology, or the study of animal behavior. Good evening. Yeah. My name is Taylor Heaton Crisologo. I'm a senior studying ecology and evolutionary biology, and tonight I'm going to speak to you about the temporal patterns of nest defense in the herring gull. To begin, by a show of hands, how many of you are parents out there? Parents, all right, parents, don't be shy. Great. So as parents, you can probably attest to the fact that across your lifetime, you have a finite amount of energy and resources which you can devote to your offspring. Now, with this finite amount of energy and resources in mind, by investing time and energy in your current offspring attempt, you may compromise your future ability to devote those resources to other future reproductive attempts. The idea being here that if you invest everything in your one current attempt, your prospects for future attempts look pretty bleak. So with this in mind, we can consider nest defense as a form of parental investment. So nest defense here demands a balance between 2 aspects of parental fitness: your investment in your current brood, but then also the potential cost for investment to future broods. So with this in mind, selection should favor a balance between your current and your future reproductive attempts. And as it turns out, nest defense behaviors do experience patterns across time. So how can we explain how parents may be optimizing investment in their offspring through changes in nest defense behaviors over time? One hypothesis, the offspring vulnerability hypothesis, posits that parents will invest most of their nest defense behavior in chicks when they are most vulnerable. At other times, when chicks are less vulnerable, parents won't invest as much. The chicks are better able to perhaps hide from predators or evade predation by running away, therefore making them less vulnerable and making the amount of energy that you need to invest as a parent less. Therefore, we would predict, under this hypothesis, that patterns in avian nest defense would change with the risk of predation that's posed to your offspring. An alternate hypothesis, the offspring age hypothesis, posits that parents will invest more effort in their offspring as the breeding season progresses and as the offspring age. Here, the idea is that as the nest season progresses, it becomes harder to renest, therefore making your current offspring more valuable to you as time goes on. Therefore, under this hypothesis, we would predict that nest defense patterns would increase with the age of the offspring over time. Before I get into my study design, though, I wanted to talk to you a bit about why I think it's important to study nest defense. Now, as you can see here in this photo, I'm wearing a helmet. I'm not wearing a helmet because I enjoy long bike rides, romantic bike rides with the gulls in the colony. I'm wearing a helmet because as a gull biologist, when you enter the colony, you face potential dive bombs, you face biting, you face just a lot of pecking and pretty intense physical behaviors from these gulls as they're defending their nests. Therefore, why would we study nest defense, in particular study the nest defense of a gull? Well, as it turns out, studies directly quantifying chick mortality are rare. Other studies have looked at predator density or the conspicuousness of chicks in the nest as proxies of offspring vulnerability. However, directly quantifying chick mortality could be a great way to test relevant hypotheses in looking at offspring vulnerability. Also, studies on larids, or gulls and terns in particular, have showed varied support. This is speculated to be because it's sometimes hard to tease apart the effects of colony defense versus the defense of individual pairs, thus making it more difficult to assess aggressive behavior. So with that in mind, I'll go over my study objectives. First, I wanted to quantify offspring vulnerability. And second, I wanted to quantify the changes in parental aggression during the nesting cycle of the herring gulls. So I was looking at a population of herring gulls that nests off the coast of Maine and New Hampshire. Gulls are colony nesting birds, and when the chicks hatch out of the egg, they're relatively immobile until they reach about 7 days of age, and then they're able to leave the nest, run around, and also potentially find good hiding places to avoid predation. Both parents participate in provisioning, brooding, and also nest defense. And for a herring gull, nest defense looks like intense physical contact, either by dive bombs, which is swooping and aiming for your head, or by bites and pecks, often paired with intense vocalizations. So the experience of a gull over its maturation looks a little bit like this. You start with this beautiful egg, which hatches into a fluffy chick that's relatively immobile until you reach that critical 7-day point, and then your chick begins to look something a little like the chick that you see in the lower right-hand corner. It looks a little more developed, it's definitely quicker on its feet, and you start to see the feathers beginning to come in. Now, after 45 days, the chick looks like it's about ready to fledge off this island. It's able to fly away, but it carries with it this beautiful mottled plumage of a fledgling bird. But after 3 to 4 years, this bird has molted into a breeding plumaged adult, completely white, and then it starts the cycle all over again. So to address my first objective, I quantified vulnerability by entering the colony and monitoring nests every day. When I was monitoring these nests, I would mark the number of eggs or chicks present, and that allowed me to get an idea of what the survival probabilities of eggs and chicks look like across the breeding season. Here, you can see a couple of my favorite pictures of gull personalities on the island. On the left-hand side, we have 2 adorable chicks, and then on the right-hand side, we have a gull that was very famous for standing on the porch and begging for food from Shoals Marine Lab students, the island that I worked at. And we all lovingly named him Peanut Butter Cookie. And he was really good at stealing food. To address my second objective, quantifying nest defense, I conducted threat simulations across 4 stages of the breeding cycle. I wanted to look at incubation, the day of hatching, or what we call chick day 1, chick day 9 of age, and then the age of chick day 25. To conduct these threat simulations, what I would do is I would have a camera set up, and then I would walk over to the nest. I would hold the egg or chick present at the nest, depending on the stage of breeding season that the egg or chick was at. and I would stand present for 60 seconds and all the while record the response of the parent gulls on camera. Because this was captured on video, I could then return later to these simulation experiments and score them on a scale of 1 to 10. So I looked at about 40 nests at the time when I had the most nests present, and I scaled them using this scale here. To explain it a little, a 1 would look like no response, so this gull is alert but not necessarily doing much. A medium response would look like some intense vocalizations and frenzied movement, but no physical contact. And a high response would look like a gull with intense vocalizations and a lot of physical contact by dive bombing or biting. And so here is an example of what a possible very aggressive gull would look like, filmed by a GoPro, not by myself. All right. Here we go. Yep, so this gull here is vocalizing and dive bombing the intruder. Oftentimes the gull strikes the target and is able to hit you in the head when it's dive bombing, hence the helmet. But if you can imagine when you walk into the colony too, it's a really kind of crazy place, but crazy fun. And now you have slow-mo too. Yeah, I'll let it play one more time too, because then you can really get the expression of pure nest defense aggression from this gull coming up on you.
Did they hit your helmet?
Yeah, they would hit our helmets and often leave little holes that looked awfully suspiciously like little peck marks. There's an angry gull for you.
Is it a GoPro or a GoPro?
We should make a new version called a GoPro that's covered in poop. So what did our results look like? Well, first we're going to look at chick mortality. We're looking at a graph that shows the predicted 5-day survival probability on the vertical axis and time on the horizontal axis. First, I'd like to direct your attention to the first point here. That looks like a very high survival probability. This is the survival probability of an egg. And it looks very high because we had relatively low losses at the egg stage for the seasons that we monitored these gulls. Moving on though. Now we're looking at the time of hatching. And then from here, it successively looks at 5-day intervals. And at the time of hatching, the survival probability drops down to about 82%. So the chances that a chick at this stage of life to survive on to the next interval is 82%, so 4 out of 5 chicks. That's pretty bleak compared to the rest of this. Whereas when you're at day 25, for example, survival probabilities are around 95%. So things are a lot more optimistic as you age as a chick. Therefore, we can construct something like a danger zone between days 1 and 10 because of the very low survival probabilities. So the take-home message here is that for the first 10 days of a herring gull chick's life, it's very hard. Now, what did it look like for the parents? Well, I'll lead you through these results step by step. So here again, on the vertical axis, we're looking at the proportion of high, medium, or low aggressive displays. On the horizontal axis, we're looking at the 4 stages where I looked at the gull behavior. Here, we're going to begin with eggs. And so eggs, you can notice that there's a relatively large proportion of low aggressive displays. However, moving on to when chicks first hatch out, you notice that there's a much larger proportion of high aggressive displays. In other words, at this time, it's really difficult to be a gull biologist in the colony because all of your birds are really kind of going nuts. Now, moving on through time, as you progress to stages day 9 of the chick's life and day 25, things start to settle down. You start to see an increase in the proportion of low aggressive displays, and again, a lot less of this high aggressive displays, so it's just cruising from there with monitoring these nests. The take-home message here is that parents defend their nest very adamantly right after the chicks hatch out of the egg. So what are some of the conclusions that we can make from this? Well, it would appear that parents are most aggressive when their chicks are most vulnerable right after that time of hatching. After about day 7, again, the chicks are gaining mobility, perhaps allowing them to better find places to hide or run away from predators. so their vulnerability decreases after that time of being able to move a lot more. Thus, this evidence supports the offspring vulnerability hypothesis, a result which I found really cool, and luckily so did my advisor, Dave Bonter. And we now have to submit this manuscript, and it's currently in review for publication in a peer-reviewed journal. Woohoo! Cross your fingers. So I'd like to conclude my presentation just with a short note about my experiences at Cornell through the Lab of Ornithology. Over the years at the lab, I've had the chance to really pour my heart into studying biology, and I had the chance to discover that I have a true passion for conducting field research. I'm really crazy about it, just ask anyone. So far in my undergrad, I've had the chance to study the breeding biology of gulls off the coast of Maine and New Hampshire. I had the chance to go down to Australia to study the awesome vocal mimicry of the superb lyrebird. And now, looking after graduation, I'm hopping on a plane to go study red-backed fairy wrens at a field site in Brisbane, working through the Webster Lab as a field technician and then later a field crew leader. And all the while, I'll be conducting preliminary data and analyses on the Albert's lyrebird, a bird which I hope to work with for conducting my PhD research. In short, I could not be more excited about my lifetime spent learning. So with that, do you have any questions? And also, I can't forget to put this slide up. I'd really like to thank my advisor, Dr. Dave Bonter. He's brought me through every stage of every process in research. When he first met me, I was a freshman who had no idea what I was doing. So thank you, Dave, and everyone else. Okay, so I'll take any questions if you have any. Sahas.
Are the individuals marked? Are males more aggressive than females or females more aggressive than males?
Yeah, so to repeat the question, we were asking if males are more aggressive than females and whether the birds were marked. So we did have individuals banded with field-readable bands. So for those birds, if we were able to take blood samples back to the lab to sex them, we knew the sex of the bird. However, that was very few of the birds that were being monitored. And unfortunately, since it's very hard to discern the sex of a herring gull, we were not able to conduct those analyses. However, if I could do it all again, I'd have all of those birds banded and know every single thing about the individuals and then look at nest events from there. But great question, thank you. Anyone else? Yes?
Along those same lines, did you know anything about the relative age of the breeding birds? Was there a difference between first-year breeder aggression versus an older, more experienced bird? Were they more aggressive or less aggressive?
Yeah, great question, thank you. So the question was whether there was a difference between aggressive behavior of younger birds versus older birds. So luckily we can tell the younger birds because although it's not too reliable, I don't think, often younger birds that come to nest will have residual spots in their breeding plumage, like on their tail feathers or on their wing feathers. We didn't really look for differences in aggression between young and older birds. However, something that has been looked at is the differences in the nest survival probabilities of younger versus older birds. That's something that I think would be more interesting to look at, because sometimes it seems like the younger birds have less luck. But no, that's not something we address. Thanks. Any other questions? Yes?
Does being aggressive work in terms of keeping your chicks alive?
Okay, so
So are the most aggressive ones doing the best at keeping their chicks alive during those stages?
yeah, and so that's something that I wanted to look at. So the question was whether more aggressive birds were more likely to have chicks that were fledging or like more successful nests. That was something that I don't think we could look at because of lower, we didn't have enough nests to really look at and compare those data. But that's a question that I really wondered about. Something else I wanted to know also is if more aggressive birds are more likely to be in the vicinity of other aggressive birds, or if wimpy birds are more likely to crowd around the very aggressive birds to kind of mooch off of those aggressive benefits. But no, unfortunately, that was a question that I could not answer. Hopefully another future gull biologist will look. Any other questions? Yes?
Were the, never mind, I'll ask it again.
Oh, it's okay. Come, we'll talk later. Yes?
Was mortality only related to predation, or were there other reasons for mortality?
I'm so glad you picked up on that. That's a great question. So the question was whether mortality was mostly due to predation or to other sources. Well, on the island, we were considering two main factors of predation being, of mortality being predation or starvation of the birds or weather effects. We didn't see much, if any at all, starving of the birds on the 3 years that I was monitoring. There we'd be looking for a decrease in the weight of the chick over time and then the chick being dead. Although we couldn't quantify it because we didn't know what was happening in the colony when I was not there monitoring. There were often times when I'd go into the colony and I'd see a great black-backed gull, the larger heterospecific that nests on the island, with the legs of my chicks sticking out of its mouth. So there was a lot of predation on the island from the great black-backed gulls. And that's what we think is accounting for most of the chick losses. Thanks. Yes. Oh, you remembered your question? Okay.
And that partially answers my question about whether there was, the herring gulls were predating on their own nests. Obviously, the greater blackback was doing some of that. Were there other birds that were doing the predation as well?
No, so we'd be looking mostly at great black-backed gulls. And herring gulls who, from my experience, won't actively go after the chicks of other birds in the nearby nests. However, the exception is when there was a territory dispute and the chicks of one nest would wander over to the territory of another nest. Then the gull parents would actively defend that nest site, and if the chick didn't get out of there quick enough, it would be killed by the other parents. Great question, thank you. Yes?
Kind of a two-part. Could you describe the most intense number 10 you experienced? And also, how would you rate the intensity of popcorn and French fry hunting compared to defending the young? They don't seem to be quite as aggressive when they're looking for food.
Okay, yeah. So 2 questions. The first one I'll address. So it was, what would be an account of the most aggressive bird? Okay, so there was 14H210. There was this nest. We monitored them by these nesting codes. I remember that. It's ingrained in my head because this bird lived in this crevice. So its nest was down in here. And so every day when I'd go to monitor that chick or egg that was at that nest, or chicks or eggs, I would have to climb down to really peek in and make sure that I was seeing who was there. But the gull parents were really smart, and they figured out that I'd have to go down there, so they'd meet me halfway, and all the while be pecking at me and squawking at me, and the other one would take turns dive bombing my head while the other was in the crevice harassing me. But they were great parents. And the second question was, why don't gulls seem like they're as aggressive when they're foraging,
Yes.
like for French fries and popcorn? My best guess would be that maybe you're seeing gulls outside of the breeding season. And so my impression is that a gull or any bird during the breeding season versus the off-breeding season are 2 very different characters. So gulls during the breeding season have a lot invested in those chicks and eggs, and so they will fight to the death for them. That said, they might also fight to the death for French fries sometimes. I don't know. Haven't observed many French fry things. Although once a gull did regurgitate a hot dog, like a full hot dog, on my friend's shoulder. And it actually bounced off. It was funny. Are there any other questions? Okay, I think that's.
Thank you, Taylor. That was great. So next up we have Sahas Barve, who is a Ph.D. candidate, also in the Ecology and Evolutionary Biology department. He is an avid birder from India and has been doing field work in the Himalayas for the past 4 years. Sahas is interested in bird conservation on mountains around the world, and according to him, he's interested in science communication and making natural history cool again. So welcome, Sahas.
Cool. Okay. So I'm going to transport you guys to the Himalayas in India, where I've been studying physiology in birds. I'm Sahas Barve. I'm in Andre Don's lab, and I'm a graduate student here at the Lab of Ornithology and in the Department of Ecology and Evolutionary Biology. So I grew up in a really tiny, quaint town called Bombay, which is inhabited by about 200,000 people. And obviously, being a birder, I needed to find a better place to bird in. So for the last few years, I've been going back to a really, really tiny village in the Himalayas with a permanent population of about 9 people and significantly better birding. But I chose this place because it's a Himalayan state, and India has about 1,300 species of birds, and this state alone, which probably takes up less than 2% of the country's total area, has about 691 species. So it has more than half of all the birds found in India, which is crazy, right? So it's the right place to be for a birder. But the question is, why are there so many birds in this state? One of the main reasons is that it's a mountainous state. And on mountains around the world, you get this sort of stacking up of species as you go up the elevation. So you have bird species that are found at different elevations, and they get stacked up. And each species thus has an upper limit and a lower limit of distribution, right? So you have, but I mean, as I said, that happens in mountains around the world. So if you go to the Adirondacks here, you have boreal chickadees in the high elevations and black-capped chickadees in the low elevations. If you go to the Western Himalayas where I worked, you have, how many is that? 6 species of sunbirds going all the way from purple sunbirds in the low elevations to fire-tailed sunbirds in the high elevations. And looks like from at least this figure, the length of the tail also goes on increasing with elevation, but I don't want to talk about that. But the thing to remember is that each bird has a distinct upper limit and a lower limit of distribution. And one of the most interesting questions in ecology has been, why is that? Why are birds limited to certain elevations? So can someone tell me what might change across the elevational gradient? What might change with elevation? Temperature, vegetation, oxygen. Great, thank you. Insects, yeah. So a bunch of different things change. And so the easy answer to this question is just a bunch of a mix of ecological and environmental variables change with elevation. So I study hypoxia physiology, or the reduction in oxygen due to reduction in partial pressure of oxygen. So it's technically not the amount of oxygen, it's the number of molecules in the air of oxygen. And I study how birds cope with what is called hypoxia. Okay, so there are some things really cool about hypoxia, and it's a constant feature around the world. So if you go to 3,000 meters anywhere in the world, the partial pressure of oxygen will be very, very, very similar to any other place in the world that is at 3,000 meters, right? So that makes it sort of a uniform constraint for anything that lives up there at those elevations. The second thing is that it's not affected by any other environmental trait, like temperature, right? So even if the air temperature is potentially 30 degrees Celsius or minus 30 degrees Celsius, the amount of hypoxia will be the same. Hypoxia has been studied in a number of human populations because evolutionarily speaking, we have this natural experiment where 3 different populations of human beings, Ethiopians, Andeans, and Tibetans, have sort of independently colonized high elevation areas around the world, and they have sort of adapted to those areas. The only sort of caveat in that is that the amount of time each population has spent at high elevations is different. So Andean populations have probably lived in the high elevations only a little more than 1,000 years, while Tibetan populations have been around for about 3,000 years. And that actually has brought about a bunch of differences in the way they cope with hypoxia. So Andeans show what is called a very lowlander response to hypoxia. So what happens in our bodies when we go to a high elevation is that to increase the transport of oxygen from the air to the blood, our bodies make more RBCs, right? They make more red blood cells, which is called the hematocrit of blood. So you increase hemoglobin concentration of the blood by increasing the hematocrit of blood. The problem with that is that the more RBCs you pack into blood, the more viscous the blood gets, right? So after a point of time, it stops being blood, it starts being more like tomato ketchup. And pumping tomato ketchup is much, much more difficult for your heart than pumping blood. So high viscosity is actually maladaptive after a certain range, right? So Andeans show this sort of a maladaptive trait where they have extremely high levels of hematocrit throughout their lives. And Andean populations actually have one of the highest rates of miscarriages in the world because pregnant women form clots very easily. As opposed to Tibetan populations, who have been around for a long time, show sea level levels of hemoglobin concentration. So their hemoglobin concentration is the same as a person at sea level. And that's because they have other ways to get around hypoxia. What they do is Tibetans have bigger lungs. So they have bigger lung capacity, but they also have nitrous oxide, which is laughing gas, in their arteries. And what nitrous oxide does is it's a vasodilator. So it keeps the blood vessel dilated and hence allows a lot of blood to pass through. So circulation is better and their lungs are better. So they can transport oxygen much more efficiently. Hypoxia has also been studied in a number of domestic and wild animals. So one of the champions of hypoxia champions of hypoxia are bar-haired geese, which are up there. Let me see if I can, yeah. So these are bar-haired geese. They are known to fly over the Himalayas. But then there are some terrestrial animals like wild yaks that have been studied, and in the Andes, llamas, and chickens have been studied a lot because chickens fail to lay eggs above 2,000 meters, I think. And people have been trying to make chickens to lay eggs because Highlanders like eggs. The reason birds are really, really cool to look at, sort of from a physiology hypoxia point of view, is two reasons. So first is that the birds have only shivering thermogenesis. So a lot of mammals have what is called brown fat, which is really, really efficient at heat production. But birds don't have brown fat and have shivering thermogenesis. So they have to shiver to produce heat in their body, right? So shivering is muscular exercise, and muscles need oxygen, right? So if you're in a cold and hypoxic place and you can only shiver to keep warm, you're in big trouble, right? The other thing is trying to fly in really hypoxic conditions. So flight requires so birds amp up their metabolic rates 10 to 12 times when they're flying. And so being able to fly in hypoxic conditions is really, really difficult. To give you a small example of how tolerant birds are, a chickadee here, consider a chickadee here in the dead of winter, it's often minus 40 degrees Celsius, the air temperature is. And a chickadee's core temperature is 40 degrees Celsius. So there's an 80 degrees Celsius gradient in probably a 2 centimeter difference from the air to the core of its heart. So birds have to really, really fire their furnace to keep warm. Okay, so let's take a segue and go to the highest point on Earth. So this is Mount Everest, and at the top of Mount Everest, the partial pressure of oxygen is only 32%, so it's about 1/3 that at sea level. Another piece of trivia is that Mount Everest is named after George Everest, so we've been pronouncing Everest wrong all the time. And old George never saw Mount Everest himself. But he was a surveyor general of the Indian. He was the Surveyor General of Survey of India, which basically made the first map of India. Mount Everest was summited by two people, Sir Edmund Hillary and Tenzing Norgay, who was not a sir for some reason. But their life histories are actually really cool, and you'll soon know why. So Edmund Hillary, let's call him Lowlander Hillary, because he was born in New Zealand and he was born in Auckland. Edmund Hillary was a beekeeper and then joined the Royal Air Force in World War II, and he was a mountaineer. So he went from sea level to the top of Mount Everest. On the other hand, Tenzing Norgay was a Highlander. So Sherpa villages are on the border of China and Tibet, so Nepal and Tibet. And so Tenzing Norgay was born in a really, really high elevation place. Like Hillary and Tenzing Norgay, we have birds. So every year, millions of birds of hundreds of species around the world travel from the low elevations in the winter to high elevations where they breed, right? So let's call all of these birds Hillary robins, okay? On the other hand, there are these birds that live at the same elevation all year round, right? So they face the same amount of hypoxia all year round, as opposed to Hillary robins that live in oxygen-rich environments for about 7 to 8 months of the year and oxygen-poor environments for 4 months of the year. So then the question is, do these two different kinds of birds, do Hillary robins and Tenzing bush tits respond to hypoxia differently, right? So what we can predict is that because most of their time is spent in oxygen-rich environments, Hillary robins will show a lowlander response, where they increase the oxygen transport by increasing the hemoglobin concentration by increasing hematocrit, right? So they will have high hemoglobin concentration and high hematocrit, which means that they have a lot of RBCs in their blood. Ketchup, yeah. On the other hand, we can predict that Tenzing bush tits that live at high elevations all year round will have some other biochemical ways to, in addition to increasing the hemoglobin concentration, to transport oxygen effectively from the air to their bodies. Okay, so my research was done in a beautiful place called Kedarnath Wildlife Sanctuary. This is a photo taken at about 3,000 meters or 10,000 feet. There are these giant rhododendron trees. So all of these are rhododendron trees that are in bloom. And it's absolutely stunning place. It's in the northwest corner of India where that star is. And we actually sampled a really, really small part of the sanctuary, and we documented more than 244 species. It's an incredibly, incredibly diverse place. Okay, but that diversity of 244 is not distributed uniformly. You have species that live at low elevation, species that live at high elevations. In the winter, there are a lot of species, about 140 species at low elevations, but only about 30 species that live at high elevations. In the summer, though, you have about 100 species that live at high elevations. And how does that happen? That happens because there are a lot of Hillary robins that migrate from southern India and the foothills to breed at these high elevations. So both these high elevation places get about 60 to 70 species that come to breed only there, right? So this is what I did. I basically sampled birds between 1,000 meters and 3,500 meters in winter and summer, and I collected data on hemoglobin concentration and hematocrit. So as you can, if you've been in the mountains, you can well understand seasons can, like the temperatures and weather conditions can change very quickly. So this is the same spot at 7 a.m. and at, I think, 9:30 a.m. or something. And to prepare for that, we have to do a lot of really, really hard training. We are constantly working out, and we are doing stretching exercises. We do a lot of yoga. And so we obviously have to do bird poses, so that's a crow, they're doing a crow pose. And some days we wake up in the morning and our mist nets are covered with snow, and we have to go out and take those mist nets down. So when it's not snowing, when we are actually working, we catch birds using mist nets like these. That's one of my technicians taking out a gray-hooded warbler, and I'm actually going to show you how we get hemoglobin samples. So I make a small incision in the bird using a needle, and then use what is called a microcuvette to take a small blood sample, and I stick that thing in a small machine that gives me the hemoglobin concentration reading. Then I use a capillary tube to draw blood to do hematocrit estimations. So you might be thinking that I'm really, really happy when I'm in the mountains, right? When there are clouds rolling by, and you can, that's a striated laughing thrush singing. But after a point of time, that becomes old. What's really, really fun is when you can hear the sound of data collection, and that's music to my ears. That's my centrifuge running, and that means I've caught birds and collecting data. Okay, so let's go back to the predictions. I had 3 basic predictions. The first one is that hemoglobin concentration within a species across its elevational distribution increases with elevation. Okay? The second one is that Hillary Robbins will show a low Lander response and hence will show high hemoglobin concentration with a correspondingly high hematocrit, okay, or volume of RBCs. The third prediction was that Tenzing bush tits will show hemoglobin concentration and hematocrit that are not correlated with one another, and which will be a sign of the high Lander response. So let's see some results. My first prediction was held true. Whether the birds were Tenzing bush tits or Hillary Robbins, like this variegated laughing thrush, the hemoglobin concentration within a species across its elevational range increased, which means that individuals of the same species at the lowest elevational distribution had low hemoglobin concentration, at the highest elevational distribution had really, really high hemoglobin concentration. So a corollary to that is that, as I had shown you before, species replace each other along the elevational gradient, right? So what we can sort of predict is that at their upper limit, the low elevation species should have a higher hemoglobin concentration, right? So here, this is a white-throated laughing thrush that goes from 1500 to 2400 meters. At its elevational upper limit, it will have a higher hemoglobin concentration than this variegated laughing thrush that replaces the white-throated laughing thrush. Does that make sense? It's a little unintuitive, but we are the first to sort of show that in multiple species. So this is we can show this with laughing thrushes and with chickadees. So these are cinereous tits and greenback tits, and across the year, the low elevation cinereous tit has a higher hemoglobin concentration than the high elevation greenback tit. My second prediction was that Hillary Robbins will have a really strong correlation between hemoglobin concentration and hematocrit, and we sort of see that with multiple species. So here each color is a different species. So many species from different families show the same reaction to hypoxia. If they are elevational migrants, they show a really tight correlation between hemoglobin and hematocrit. As opposed to Tenzing bush tits, which I predicted to have a relationship like that, and the real data also showed the same thing. So Tenzing bush tits or resident birds don't have a strong correlation between hemoglobin and hematocrit. So then I sort of decided to throw a slightly more complicated statistics at it, and did what is called linear mix modeling approach. And I'm not going to get into the details, but basically what it says is that hemoglobin concentration is driven by the hematocrit of the bird and whether the bird is an elevation migrant or not, which means whether the bird is a Tenzing bush tit or a Hillary robin greatly drives the hemoglobin concentration of a bird. Okay, so why is this important, right? Why should we care about physiology of birds other than just from a scientific point of view? So one of the important things is we really don't know how physiology affects other interactions like competition, right? So we can say that at its elevational upper limit, a species might be physiologically stressed, but does that sort of translate into how competitive it is to other species, right? So I'm going to show you a small video of that Shelly Shah, another undergrad here, took of a greenback tit and a cinereous tit at a feeder in the Himalayas. So you might actually hear the sights and sounds of a Himalayan village. So that's a greenback tit. So that's a greenback tit feeding on a feeder, and that's a cinereous tit. So cinereous tit is the physiologically stressed underdog, and the greenback tit is the overlord of the feeders right now. And you're going to see what happens when a cinereous tit tries to steal some feeds from the greenback tit. So cinereous tit makes its way slowly. Yeah, so the greenback tit does not like it, and cinereous tits are complete pushovers at their elevational upper limit because they get outcompeted potentially because they're physiologically stressed and can't be good competitors. The other reason, hold on, let me go back to my presentation. The other reason why studying hypoxia physiology is really important is that if you plot all the species of birds on a world map, you can actually see the outlines of all the major mountain systems. So you can actually see the Himalayas, the Andes, the Central African Rift Valley mountains. They all light up because mountains around the world are packed with bird species. And we don't know anything about how these birds respond to hypoxia, right? So one of our major predictions is that with climate change, species are going to move up slope as the temperatures rise. But if they can't cope with hypoxia, they might not be able to move up slope as readily as we thought they could. And the last reason is that we are losing birds really, really quickly. So this is a photo from my field site. In a square foot of mud, you have a pug mark of a leopard, tire tracks from a car. These are the hoof prints of a cow, and someone had scribbled, "Anu, I hate you." And all this happened in 1 square foot of mud. So around the world, on mountains, there's a lot of habitat changes that are happening. And we are losing a lot of really, really cool birds, or many cool birds are getting endangered because of habitat loss. And we might lose a lot of those species before we even know anything about them completely. So with that, I would like to thank my several funding agencies and my hardworking field assistants, and leave you with a few photos of Himalayan birds. Thank you. Are there questions? Yes.
You had a map showing the distribution,
Mm-hmm.
and your focus was on mountains.
Right.
I saw very little difference in the Rocky Mountains, for instance. Is it primarily tropical mountain ranges that are the high habitat range, not northern mountains?
Right. So yeah, the question is if tropical mountains have more species, and yes, the answer is yes. Tropical mountains are sort of have disproportionately more species than other mountains around the world. Yeah. Yes.
Do you have a sense of whether there are behavioral changes that they can make as well to cope with hypoxia in terms of like changing activity level and things like that?
Nothing like that is known. So we know some birds will go into torpor at night to sort of reduce bodily functions, but there is very little they can do to sort of overcome hypoxia behaviorally. Yes, Tom.
Can you speculate how the Highlander birds adjust to their altitude? What mechanism?
Yeah, so they might have, just talking from a sort of a blood perspective, they could have smaller, so this has been seen in deer mice in the Rockies. They have smaller erythrocytes, so they have smaller RBCs, which increases the total surface area to volume ratio for binding with oxygen. But they might also have, since you're a physiologist, I can tell you, they might also have what is called as MCHC, right? So they have more hemoglobin packed into each red blood cell than your normal red blood cells. So those are the two sort of blood-related ones. But then there could be, they could have bigger air sacs, or I don't know. There could be other mechanisms also. Yes.
So for the Hillary type birds,
Mm-hmm.
within their range, the bottom of it or the top of it on the mountain,
Mm-hmm.
is there any reason why, I mean, can you tell, do the younger birds go for the lower level and the older birds the higher level, or is there some benefit to being higher or lower within that bracket?
I don't.
Is the survival different maybe?
Right, that's a great question actually. I don't know if there is any age, if they sort of sort themselves out age-wise. But what I can say is that the nesting season is longer in the lower elevations than at the highest elevations, because it's even a span of 1,000 meters on a mountain makes a big difference. You can potentially get 15 or 20 days more to nest in the low elevations as compared to high elevations. Yes,
Are
Caleb.
you measuring the percentage of hemoglobin inside the red blood cells?
No, but if you can measure the size of the red blood cell, and if you have the hemoglobin concentration of the bird, then you can get at what that is pretty well.
Are you getting that or you're not going to do that?
Not in the Himalayan birds, because to measure that, you need to get the blood under a microscope with a camera within 24 hours, and that's something that I can't do in the Himalayas. Yeah.
Do you expect that there's a difference in the incubation period? Because oxygen levels are very important in the incubation period.
Yeah, so it's been shown with ptarmigans in the Rockies that, so ptarmigan eggs have bigger pores, and the incubation period is longer in ptarmigans. I don't know if people have looked at incubation periods in songbirds in the Himalayas or actually anywhere, but it has been shown with birds in the Rockies. Yes. Cool. Well, thank you so much.
All right, thank you, Sahas. So last up tonight, we have Connor Taff. Connor is a postdoctoral associate here at the lab. And before coming to Cornell in 2015, he completed an NSF-funded PhD and a USDA-funded postdoctoral research at University of California, Davis. Connor's been studying wild birds for over 10 years, and during that time, his work has addressed questions about breeding biology, life history trade-offs, sexual signal elevation, epidemiology, and movement ecology in common yellowthroats, greater sage-grouse, and American crows. Connor is also the recipient of multiple awards, including the Cooper Ornithological Society Young Professional Award and the UC Davis Merton Love Award for Best Dissertation in any area of ecology or evolution at UC Davis. So we're lucky to have him. Welcome, Connor.
Thanks. All right, so thanks for giving me the opportunity to talk to you tonight. So I want to start by showing you this picture. And One of the things that I find most fascinating about studying wild animals and that got me excited about pursuing this as a career is the amazing degree of diversity that you see in signaling traits across a variety of species. And so that's true certainly in visual traits like what we're looking at here, like dewlaps on lizards and stalk-eyed fly antennas, and of course in plumage patches in birds. And understanding how and why these elaborate traits have come to be has been a real puzzle for biologists for a long time. So as many of you probably know, Darwin proposed about 150 years ago the mechanism of natural selection as the main driver of evolution, but he himself was really puzzled by how these elaborate traits that seem to serve no utilitarian purpose could evolve under natural selection. And he thought a lot after his first book came out about how we could see this evolution, and was so puzzled by it that he wrote to his friend Asa Gray in a letter in 1860 that small trifling particulars of structure often make me very uncomfortable. The sight of a feather in a peacock's tail whenever I gaze at it makes me sick. And a lot of his career was spent thinking about these sexually selected traits, and the way that he eventually resolved this paradox to his own satisfaction was by proposing a special type of natural selection that he called sexual selection. And basically he argued that in addition to competing to survive and reproduce, animals might compete for access to mating opportunities. And there were two main ways he thought this could happen. One being intrasexual selection, where members of one sex compete with each other for access to the opposite sex. So things like bighorn sheep, where males butt heads and only the most dominant male can mate with a harem. And the other being intersexual selection, where members of one sex have a mating preference based on some aspect of the signaling traits of the opposite sex. And this is where he thought you could get the evolution of these elaborate plumage patches when females, we often think about females, although there are some species where these sex roles are reversed. So typically females exert some kind of mating preference, and that over many generations leads to the evolution of these truly elaborate traits that I showed you in the last slide. And so it's relatively easy to imagine in birds how this might produce those signals in some species. So things like greater sage grouse, where you have a lecking mating system. So these are the birds that my advisor worked on, and I did a couple field seasons on them. And they have these amazing mating system where every breeding season the males gather on a lek. They perform this really complex behavioral display with vocalizations using these inflated air sacs. And they also have this elaborate plumage with plumes on top and this great spiky tail in the back. And in a typical sage grouse lek with 100 displaying males, you might only have 3 or 4 males who successfully mate the entire season. So females are coming and visiting the lek, and they basically have free choice of any male that they want to mate with. And the most successful male might mate 40 or 50 times. So you can imagine that if the degree of elaboration of this plumage is related to the choices that these females are making, you can get really strong selection leading to more and more elaborate plumage. But the reality for a lot of birds turns out to be that about 90% of the birds in the world are socially monogamous. So they don't have this elaborate lek mating system. Instead, like a lot of the birds around here, they're territorial, where males have a single territory. They often have 1 or maybe 2 females who settle on that territory. And so you don't have this apparently very large reproductive skew where a few males are monopolizing all of the matings. And so it was a puzzle for a long time to understand how in these socially monogamous species you could still get fairly elaborate plumage. And so this is the bird that I did my dissertation on, Common Yellowthroat, that I've been working on for over 10 years now. They're maybe not quite as elaborate as those sage grouse on the last slide, but they do have this really nice striking plumage. So the males have this black facial mask that the females don't have at all. And they also have this very bright yellow bib that extends down their whole front in some birds. And females do have a version of the yellow bib, but it's smaller and duller. And so about 30 years ago, the key insight in understanding how we could get sexual selection acting on plumage in these socially monogamous species occurred about 30 years ago when it became possible to apply genetic paternity tests to wild birds. And it turned out that even though we have these socially monogamous mating systems, in most cases, the genetic mating system is actually not monogamous. So even though you have a territorial male and a female defending a territory together and provisioning the young together, some of the nestlings in the nest of most songbird species are actually sired by males on neighboring territories. And in some species, this can be a really extreme. So in some socially monogamous species, you might have actually 50 or 60% of the nestlings sired by males on nearby territories. And so in yellowthroats, the kind of main questions that we've been addressing over the course of this research program are, what are these signals actually used for? So are they related to reproductive success? Are females paying attention to them? What information is conveyed by signals? So if females are paying attention, is there a reason? When they pay attention to these signals in mate choice, are they getting information about the underlying physiology of those males? And how is honesty maintained? So the kind of flip side to that is, are there physiological constraints that prevent males from cheating? If it's good to be bright and have a big yellow bright bib, why don't all males have big yellow bright bibs? Is there something that's creating variation in the population? And so before I go into some of the results that we found, I just want to give you a little bit of natural history on common yellowthroats. So as their name implies, they are really common. They're found over pretty much all of North America. So the breeding range here in pink is pretty much over the entire northern United States and Canada. They also have some resident populations that don't migrate in Florida and California. One of the things that I really like about yellowthroats is that they're a bird that is super common, they're very abundant, but a lot of people who aren't birders have actually never seen one. In about a month, if you go out with your car windows, I think many of you who are listening to this probably have seen yellowthroats, but if you go out driving around pretty much any of the areas in their range with your windows down and you're near a swamp, you'll hear them singing from the swamp. So they're really prolific. This is what the typical habitat looks like. This is a picture of one of my field sites. So this is a series of beaver meadows that extend up an old river. And the main field site that I've worked at is here in Saratoga Springs, New York, so it's only a couple hours away from here. Pretty similar to the habitat we have right around Ithaca. So each field season, we go out and on the territories that we're monitoring, we catch all the birds in mist nets. So you can see us taking a bird out of a mist net here. We catch all the adult males and females and take your standard kind of morphological measurements of the body size of these birds. We put these colored plastic leg bands on. And we put a unique combination on each bird so that we can then go out with binoculars and identify them for behavioral observations later. And we take blood samples from all of the adults and nestlings in the population so that we can do the genetic paternity analysis, and also for some physiological measures looking at the blood. But the vast majority of our field season is actually spent tromping through swamps in knee-high boots trying to find these nests. So the females in particular are incredibly cryptic. They're in this dense marshy habitat. And here's a female sitting on her nest here. So you can see she doesn't have the black facial mask. And her back in particular just blends in perfectly with the vegetation that's in these swamps. And so this bottom picture here is showing you a yellowthroat nest from about a foot away. So you can imagine that these, they almost, when they're building their nest, they're almost like mice. They get a strip of grass and they run along the ground to their nest and run along the ground away from their nest. So it's very hard to follow them. They're very skittish if you get too close to the nest. And they have high predation rates, so we end up having to find the nest over and over again. When we do find them, we monitor the nest every day until they hatch. So this is a yellowthroat nestling that's just a couple of hours old. And then when the nestlings are 5 days old, which they are in this bottom picture here, we go back and put bands on them and take blood samples from the nestlings as well. And so with the help of a lot of people over many years, we've built up this large data set. Okay, so before that slide, we also are interested in a couple different plumage measures, and I just want to show you what those look like here. So first, I mentioned that males have this black facial mask. It turns out that this varies a lot between males. So these are two males in the population, one with a large mask and one with a small mask. And we photograph all the birds against these standardized grid backgrounds, and then we can use a computer program to measure the area of those plumage patches later on. They also vary a lot in bib. So again, this male on the left has a large bib. This male on the right has a small bib. We measure the area of the bib as well. And for the bib, we also measure the variation in the quality of the color that's in the feathers. And to do that, we pluck a few feathers from the center of the bib and use something called reflectance spectrophotometry to generate these reflectance spectra. And so the x-axis here is wavelength in the visible spectrum. The y-axis is the percent reflectance, so that's how much reflectance is at that wavelength. The yellowthroats have this is a typical profile for a male where they have a peak down here in the UV range, which we actually can't see at all, but birds, because they have a fourth color cone, can see. So presumably these patches actually look quite different to a female yellowthroat than they do to us, but we can quantify the variation in that yellow coloration. They have a dip in the middle wavelengths and then another big peak in the yellow range, which is what we see when we look at these birds. So they appear yellow to us. They also have a single song that they repeat over and over again throughout the breeding season, and so they don't have a repertoire, but each male has their own song. So these are the chips. It's kind of a contact chip. And that's the yellowthroat song. So it's witchety, witchety, witchety. They're really prolific singers, and they're the most common singers in this habitat. And I'll come back to that in a minute. So here's the slide I meant to be at before. So over the last 10 years, with the help of a lot of field assistants and a lot of effort, we've built up this big data set from our local field site where we've followed a little over 200 males, about 150 females. We found 500 of those nests, which I think is probably the major accomplishment of this entire project, and sampled about 800 nestlings. And one of the nice features of our field site is we have these swamps that are surrounded by dry woodland on all sides. So we don't have any neighboring territories of yellowthroats that we're not following. So we're actually able to assign genetic paternity to all of those 800 nestlings. And it turns out that 18% of them are extra-pair young. So we have this potential mechanism for sexual selection through extra-pair mating success in addition to within-pair mating success. And I'll just give you a quick rundown of kind of the early results from our project. The basic takeaway is that we know that plumage size and coloration are related to a lot of things that seem to be important for mating success. And particularly bib coloration and bib size seem to be more important than mask in our population. So different aspects of bib size and coloration are related to male dominance and female preferences when we do aviary trials. In our natural population, both extra-pair mating success and within-pair mating success are related to bib coloration. And there's also relationships with survivorship. So plumage coloration predicts survivorship over the winter. And so you can imagine that a female paying attention to bib coloration might be interested in survivorship to the extent that that's a heritable trait that their offspring will then get. Similarly for song, so we haven't done quite as much work on song, but we do know that extra-pair reproductive success is related to song performance. So males that sing more consistently are able to sire more extra-pair young. And again, there are relationships with survival. So it seems like there's a potential for selection driving the evolution of some of these traits in our system. But I want to talk a little bit more in a little bit more detail about some work we've been doing more recently. And first I want to talk about a project that we've been doing to try to look at communication in networks in our population. So the early work that we've done has all really focused on what was going on between a male and a female on their home territory. But yellowthroats, like most songbirds, or a lot of songbirds around here, have this network of territories where at our field site you have these pairs of interacting males and females, so the male on the left here, female without the mask on the right. So you can think of this as a territorial pair. We also have some males in our population who come, claim a territory, sing all season, and never attract a female. Those tend to be the young males, but we do put them in here as well. And when you have this network of territories, you have a potential for communication to occur not just between a male and a female on their own territory, but at this larger network level. And that's maybe not as true for something like plumage that has to be assessed at close range, but for song that carries a long distance, there's a potential to both signal to neighbors and to listen to what's going on neighboring territories. And so we've been interested in trying to understand, first of all, whether males, when they're making their singing decisions, are even paying attention to the neighborhood context, and whether they're changing their singing to match kind of subtle changes in what's going on in the entire neighborhood rather than just on their own territory. And so we've been doing that using remote monitoring of singing behavior. So we use these great waterproof and weatherproof song recording devices that record to SD memory cards. They can be set to record on a fixed schedule, so we can record many territories at a time and for many days in a row, and get total song production from all of the males in our population. As I said, males don't have a song repertoire, so we're actually able to use recordings that we made using a microphone of each male to match up to these and identify each of our focal males to see how many times they were singing. And I just want to give you a snippet of what this would sound like. So if you were at one of my field sites in upstate New York in early May, just before dawn, you hear the spring peepers going here. And before the spring peepers have even stopped, you hear the first yellowthroat come in. So you can see in the spectrogram here, yellowthroat vocalization. These recordings do pick up a lot of other birds, things like gray catbirds, which are really amazing singers, but are actually the most annoying birds in these recordings because they just sing incessantly and make it hard for us to measure what we want. A veery here, that's one of my favorites, singing from the edge of the swamp, and a song sparrow. But in these habitats and in these recordings, it's quite amazing. Actually, the yellowthroats are by far the most prolific singers. And so this is an early season recording. You actually can hear 3 males counter-singing at the edges of their territory here. And so they're probably setting up the territory boundaries early in the season. And at the end here, one male intrudes into the focal male's territory, and you'll hear a rattle, which you can see here. So that's the most aggressive yellowthroat vocalization. That's that focal male chasing that intruder back out of the territory. And so these recordings can make these great soundscape audio files that if anybody is interested in downloading some to listen to while you're going to sleep, I have thousands of hours of these recordings. But you can also pull a lot of interesting data out of them. So with some signal processing, using a computer program and a lot of manual checking, we can actually pull out the total song production for all the males in our population. And we have really detailed census data from all of these years, so we know exactly what was going on in every territory. And we can look at how males are actually changing their singing as the fertility status of neighboring females changes. We know, and these recordings confirm, that males decrease their singing a lot when their own female is fertile, presumably because they're following the female around, trying to mate with her and stop other males from mating with her. But we haven't known before this what males are doing in response to neighboring females. And it turns out, I'll just jump to the results here, that on the y-axis here, you're looking at the total amount of song production. The x-axis is the number of fertile females within 400 meters. And we know from some previous work that 400 meters is about the maximum distance that males will travel to sire extra pair young. So for each additional female within 400 meters, males are singing about 10 to 20% more each day. And these birds weigh 10 grams, so they're about the size of 2 U.S. quarters, about the weight of 2 U.S. quarters. And they're singing in some of these recordings up to 2,000 songs in a day. So it might not seem like a huge increase, but 10 to 20% increase for such a small bird that's already singing a lot and might have 3 or 4 fertile females in the neighborhood is a pretty substantial increase in singing effort. And I think one of the things that I'm most excited about with these results is just that it suggests that these males are, these small songbirds are aware to some extent of what's going on, not just on their own territory, but on these neighboring territories up to 400 meters away, and actually responding to that in a way that seems to make sense adaptively. We don't know exactly how males are getting that information. One possibility that's intriguing is that they're actually paying attention to how much their rivals are singing, and as their rivals decrease their singing, the focal males are actually ramping their singing up because they know that there's a fertile female in that territory. Another major focus of our work, and I just want to touch on this briefly, has been trying to link physiological mechanisms to the expression of signals that we see and ask whether physiology in some sense might be constraining the expression of ornamental traits. And so we've done this in a number of different ways. I just want to mention one here as an example that we've done recently, and that's that we've looked at telomere shortening with respect to ornamentation. So telomeres are these repeated base pair sequences that cap the ends of eukaryotic chromosomes, and they shorten throughout life as cells divide. They've received a lot of attention actually in human medical literature. They're implicated in a lot of cancer formation, and a lot of cancer biologists work on telomere erosion. They're thought to be related to the aging process, and so we've measured for our birds where we have consecutive years of sampling, we've measured telomere lengths between years and ornamentation between years, and it turns out that on the y-axis here, you're now looking at the absolute telomere length. The x-axis is coloration, and it turns out that coloration in a male's first year of breeding predicts their telomere length during that breeding season and during the subsequent 2 breeding seasons. And The change in telomere length between years is also predicted by coloration. So males that have bigger, brighter yellow bibs have longer telomeres and are losing telomeres at a slower rate. So again, this suggests to us that males paying attention, or females paying attention to ornamentation are potentially getting information about the underlying physiology of those mates. And then the last thing that I want to talk about is something that I don't really have results from yet, but I'm excited about, we've been doing over the last couple of years. And I'm not really working actively on yellowthroats right now, but we have some side projects still continuing. And one of those is that we've been looking at the isotopic signature in feathers to try to get a sense of where males are moving across the landscape. So everything that we've done so far has really been focused on this one field site in upstate New York. But we're trying to scale this up to understand how movement across the landscape might influence the expression of signals that we see in our breeding population. And there's this nice feature of precipitation that as you move northward in latitude, the ratio of hydrogen to deuterium, the heavy isotopic form of hydrogen, changes predictably. And because feathers are inert tissue, once they're grown, that ratio is locked in. So when we sample feathers, those are feathers that were molted the previous September. We can put them in a mass spectrometer and look at this ratio and actually know where the birds, a fair approximation of where the birds are when they grew those feathers. And it turns out that the latitude where feathers were grown is related to coloration in our birds among yearlings. And what seems to be driving this is that some birds in our population were hatched farther south from our site and are actually dispersing up into our site as breeders. And so there's potentially some interesting things going on here with the ecological drivers of coloration leading to variation in our field site. And the flip side to that is that we'd also like to know about where our birds are spending the winter. And up until now, we don't have any information on where they've been going. Despite the many, many years of people banding hundreds of thousands of birds on the breeding grounds in North America, resighting those birds on the wintering grounds is vanishingly rare. So we often don't know. We know the yellowthroat range in the south, but we don't know where our particular birds spend their winter. And yellowthroats, as I said, weigh about the weight of 2 U.S. quarters. So until very recently, it's been impossible to put tracking devices on them. But in the last couple of years, it's become possible to put these geolocators that weigh less than half a gram on birds as small as yellowthroats. And so we actually put 20 of these out last year. And the way that these work is it's basically just a very small battery, a memory card, and a light sensor. And it records the time of sunrise and the length of daylight. And with those two pieces of information, you can actually calculate latitude and longitude. So we put 20 of those out last year, and in about 6 weeks, or actually right about now, those birds, presuming they're still alive, should be starting to think about migrating north. In about 6 weeks, we should know if any of them return. We can catch those birds again and pull those off, and for the first time get information on where our birds are spending the winter. And I just want to acknowledge that what I've talked about here represents a decade of work with a lot of collaborators, and so there's a whole list of mostly undergraduate field and lab assistants who've worked on this over the years, funding sources that have paid for various parts of the project, and collaborators and advisors who've helped me with various parts of it. And I'm happy to take some questions. Yeah.
I'm having trouble reading your graph. Does it suggest that 3 is the ideal number of outside females to generate song? Because it looks like it drops off when you get up to 5 or 6.
Yeah, it looks like it drops off, but so the question was in the graph, the relationship between the number of fertile females and song production, why does it look like it drops off when you get up to 5 and 6 females? The answer really is just that there are very few instances where there were 5 or 6 fertile females. So the sample sizes for those two are very small. Yeah, so whether they would keep singing more, if there were more fertile females, we don't know what the maximum is there. Yeah.
Is there a correlation between, on the amount of bib size, on the bib size, the more there is in the visible light, does that correspond to an increase also in the UV?
Yeah, it does tend to be. So the way that the feathers are actually grown, it's interesting, and we haven't fully quantified this, but the feathers themselves, if there's no pigment deposited in them, are white. And so the elevation of that full reflectance curve has to do with how pure the white coloration is. That's not a pigment, it's a structural color. And then once you have that pure white feather, as it's growing, they deposit carotenoids, which is the pigment that actually turns it yellow. And the carotenoids are actually a subtractive pigment. So they pull that trough in the middle down. The more carotenoids you have, the lower that trough goes. So that initial shape, there is a correlation between the UV coloration and the yellow coloration. That kind of whole elevation of the shape is probably determined by the actual structure of the feathers rather than the pigments. And it's possible, I mean, we haven't done this with our birds, but it's possible to actually measure the coloration and then do a process where you pull all of the pigmentation out and then remeasure the white coloration and get how much pigment was actually deposited there and whether there's variation in the base white color as well. Yeah?
Let me see if I can get this right. So, a lot of this, most of this was done in relation to the extra pair sexual bonding.
Mm-hmm.
Is there a reason why, or that's the question, does any of this coloration advertising have anything to do with the original pairing, their social pairing? Is there an issue there of attraction that's important at that point?
Yeah. So the question is whether, I was mainly talking about extra pair mating success, the question is whether there are relationships within pair mating as well, and there are some, particularly, so I mentioned that for the first-time breeders, there's a substantial minority of males who have a territory but never attract a female at all, and those tend to be the first-time breeders who have the smallest, dullest bibs. So in that first-year group, there's actually a lot of variation driven by just whether you get a female or not. And a lot of that is determined by bib size. Among the older birds, almost all of them, I think maybe 149 out of 150 older birds that we've looked at have attracted a female. So there's not a lot of variation there within pair. There is some variation in how many eggs the females lay, and there's some relationships with that, but it's not as strong, and there's not that much variation because they can lay 3, 4, or 5 eggs, and almost all of them lay 4 eggs. So there's not a lot of variation in terms of total reproductive success. There might be something going on. I mean, one thing that we don't really have a good handle on, all the nestlings that we banned, so all those 800 nestlings that we banned, they leave our site, they migrate south, and we never see them again. So they don't return to our site. They return probably nearby our site. Actually, I think we saw 3 again that came back into our site. So it's possible that there's something going on with the quality of the offspring where the within pair mating might be important for determining whether offspring have a high chance of surviving to reproduce as adults, but we don't have survival data for the nestlings. We just know how many fledged at the end of the season. Yeah.
I was really amazed at your data collection, the methodologies you used to get at all this data. It was amazing. But we were told as kids never to get close to a nest, never to touch a little bird, never to even breathe on it or look at it because it'll abandon the nest. Is that all just nonsense that we were told?
Yeah, I mean, for the most part, birds, I mean, that's definitely more of a mammal thing than a bird thing because the birds, well, this has been challenged a bit, but the classic view is that birds don't really use olfaction, and so touching the bird is not going to do anything in terms of, putting your scent onto the bird is where that, typically when people talk about that, they think about putting your scent onto the bird. Now, if you're disturbing the nests, I mean, you can cause abandonment, and we're very careful when we're out there when we find the nest, but when we're doing our nest checks, we do them as infrequently as possible, and we'll try, so people have done experiments on this where if you walk to a nest and make a path, predators like weasels and things will actually follow the path that you make to find the nest. So we'll do things like taking, big steps over and leaning over with a stick to look in and say, okay, that nest is okay for today. Except for on the day when we actually go band, we'll try to be as minimally invasive as possible. Okay, thank you.